Hello, everyone. I'm Alex, and my 198B project will be on a ultrasound investigation on closed head injuries. So let's get into it. Traumatic brain injury is one of the leading causes of mortality stemming from blunt head trauma. It manifests as cognition deficits, motor ability deficits, sensory ability deficits, and can even result in coma or death. A substrate of reference for consequences of TBI is needed to quantitatively assess the effects of head trauma. So for this experiment, we're going to be using functional connectivity perturbation. Functional connectivity can be thought as the phenomenon where we see two or more brain regions exhibit activity in tandem. We can thus inference that these two regions are related in one way or another to assist each other with executing a specific task or function. In terms of the physics of TBI, TBI can be separated into two aspects, bulk and shear modulus. Bulk can be thought of the linear impact analogous to blunt head trauma, whereas shear modulus can be thought of like rotational impact analogous to like whiplash from a car crash. Now, a paper that's been heavily implicated in this type of research is in Cleveland et al., published in 2013 by Frontiers. In that paper, it says that brain tissue is about 5 times more sensitive to shear stress than bulk stress. Now, here's the protocol schedule that my project is going to be taking. The black boxes indicate days where anesthesia and impacts occur, and the red arrows indicate days where imaging occurs. Days 2 and 7 are the only days where anesthesia, impact, and imaging all occur. All mice undergo head bar surgery, and they're given 24 hours recovery time. And then they were split into one of 3 groups. We will impact them with a CCI machine according to the classification of the group. The top-down group receives impact at the bregma of the junction between 2 frontal bones and parietal bones. The lateral group receives an impact between the lateral angle of the eye and the anterior part of their ear. And then the sham group doesn't receive any impact, but they're still anesthetized. The lateral CHI and top-down CHI groups experience impact by electromagnetically controlled piston. The impact speed for both groups was around 4 meters per second. All mice were then given 30 minutes to wake up and recover before undergoing functional ultrasound imaging, and then images were then inputted into MATLAB to obtain functional connectivity perturbation networks. So our hypothesis is, since brain tissue is 5 times more sensitive to shear force than bulk force, if we apply the same amount of force for a lateral CHI group and a top-down CHI group, we should observe more perturbation in functional connectivity at a quicker progression rate in the lateral CHI group exhibited and collected through functional ultrasound imaging. Our first figure shows that there is a significant difference in functional connectivity change between lateral and top-down CHI groups. We compared all mice from all time points to identify gross functional connectivity network perturbations between lateral and top-down mice, all versus sham mice, so compared. We observed significant hypoconnectivity in both lateral and top-down groups when compared with the control group. The perturbed network in the lateral group was significantly larger and encompassed more brain regions than the top-down group, providing support for the claim that trauma directed from the lateral side perhaps could result in a more severe form of TBI. This is kind of consistent with previous literature, as the most common fatal head injuries, such as subdural hematomas and diffuse axonal injuries, are more easily caused by rotational kinematics and shear stress. Now, we're also interested in the dynamic progression of functional connectivity network perturbation over time. Now, progression was then assessed by eliminating the baseline brain activity from the brain activity result recorded at the 1-hit and 5-hit time points. The resulting matrix reflects the dynamical change of the brain between the 2 time points. So as you can see, for the top part, it's assessing the dynamical change in connectivity between baseline and 1 hit. The comparisons were conducted to identify progression. Now, results showed that lateral mice exhibited a significant decrease in functional connectivity compared to the sham group. On the other hand, comparisons between the top-down and sham groups showed a relative increase in functional connectivity. Of note, there were a greater amount of brain regions implicated in network perturbation for the lateral group compared to the top-down group, suggesting that lateral rotational trauma could result in a more aggressive progression of functional connectivity change in the same amount of time when compared to the top-down group. Interestingly, perturbed networks in both comparisons heavily implicate the ventral pallidum as a region of interest, which harbors many projections to other regions of the brain. Now, this could be an interesting focus of like a little offshoot study. Additionally, the relative severity of lateral trauma is further confirmed when analyzing the difference between images taken at baseline and images taken 1 week later after 5 hits have been applied. Results revealed a significantly more perturbed network, including both increases and decreases in functional activity between brain nodes in the lateral mice compared to control mice, suggesting further progression beyond the images taken at just 1 hit. On the other hand, top-down mice interestingly showed a much smaller perturbed network compared to the network at 1 hit, indicating that there could be potentially some underlying recovery mechanisms due to the lack of traumatic severity. I would like to do some acknowledgements for this presentation. My PI for my lab is Dr. Neil Harris. My 2 postdocs are Dr. Ashwin Pedar and Stefano Lepore. And then I've also worked in this project with another undergrad, Jackson Algashev. And this entire lab is funded by the David Geffen School of Medicine at UCLA and the BRI within the Department of Neurosurgery and Neurobiology. And I personally have received funding from the Life Science Dean's Award for Neuroscience Research. Thank you, and hope you enjoyed my presentation.